Hello dear friends, you are welcome to my YouTube channel Pharma Learning. Today I Dr. Shamsher Singh welcome you all and I am going to discuss about toxicological studies, that why there is a need of toxicological data and why we perform toxicity studies on animals like rodents and non-rodents. What are the basic principles of toxicological studies and what are the basic steps to be followed whenever you will do the toxicological studies. So as you know that this toxicity studies is very important because whenever anybody want to go for clinical trial analysis, before clinical trial initiation, there is a requirement of data which is called as toxicological data. So that means it include both toxicokinetic and it will include both toxicodynamic. So these type of studies, toxicity studies, which are to be performed on rodents as well as non-rodents. So in the previous lectures, you can see that I have explained that why we consider both rodents as well as non-rodents for the toxicity studies. Because our aim is to find out LD50 value, that is lethal dose. So it means the dose at which there will be no NOEL, no observed effect level. So that means we always go for two type of dose analysis. Either it can be a fixed dose design. Suppose you are taking 5, 50, 500, 5000 like that. So this is called as a fixed dose design analysis because during toxicity studies we think number of factors are to be involved. I will discuss in this lecture with you so that you can understand number of the things which are to be required whenever we go for the toxicity analysis. The first thing, we always do toxicity studies on animals and we assume that if we didn't found any type of ADR or a side effect on the animal related to drug, hope we considered it will be safe to be utilized in the clinical trial. The first thing. The second thing, whenever we design a short-term toxicity studies, acute type, a medium, subacute type and a long-term chronic type, we assume that these number of days or the number of weeks for which we are doing the toxicity studies in rodents are equivalent to humans, because we always give some of the drugs to the human for 3 weeks, for 1 week, for 2 months, 3 months, 1 year, like that. So depending upon that, if that drug firstly will be exposed toward the animal, the chances of the adverse effect on the human being will be less and we will generate a safe data about the drug. So I am going to discuss with you that there are basically three type of toxicity studies which we perform on the animals. Now, how to select the animals, which type of animal to be selected, which type of animal are not to be selected in some conditions, I will discuss this thing in the coming lecture. So in this lecture I will discuss with you about the basic principles, that how we can follow OECD guidelines and ICH guideline, because this toxicity studies is conducted under 21 CFR part 314. So that means this is the part of the you can say this particular analysis where we do the toxicity studies. So this code of federation, that means 21st 21 CFR, and this is considered under a part 314. So this is the particular section where we do the toxicity studies to move our drug for the clinical analysis. So if we go back to the some of the available data, the toxicity studies has been performed by Paracelsus, and afterward around in 1920 the animal studies was initiated to find out toxic effect of the drugs. So that's why in America and in some other countries, likely there was a one of the member of Jacob. He was a scientist in the FDA, US FDA. He also did a toxicity studies, and he preferred at that time that rabbit will be a useful animal for the eye irritations. So that's why from 1960, by this Jacob, who was a scientist in US FDA, he established one of this model that rabbit can be used to perform eye irritation. So from that date, from 1960 to till date, this eye irritation test is performed on the animals. So remaining which type of animal are to be preferred, I will discuss with you after this lecture. So you like, subscribe this channel so that you can get a lot of knowledge on this toxicity studies, basic research, how we can do research on animals, zebrafish like that. So there are few type of the toxicity studies. Acute, as it indicate that the data will be analyzed, the drug will be administered or the drug will be applied for 7 to 28 days, at least for a duration of 1 month. The second is subchronic toxicity. It means the duration will be for 3 to 6 months. And there are few these type of toxicity. I will in the next coming lecture I will explain each and everything about this. Now, chronic and long-term toxicity, it means it could be for 12 months, and if it is a long-term toxicity like carcinogenicity, mutagenicity, so it will be from 18 to 24 months, like 2 years. So how you can see that this is a animal and we are administering a one molecule to animal. You can see that this molecule or a drug, it is producing a lot of changes in the body of that animal. It can be due to some organ deposition, it can be due to some organ damage, it could cause some of harmful effect on the mind, that is CNS associated toxicity, cardiovascular associated toxicity, carcinogenicity, mutagenicity, reproductive toxicity. So that means all these parameters are to be required whenever you will go for new drug application, you can say for the clinical trial analysis. So what are the basic principle? This is very important for the young researchers so that they understand that how you can do toxicity by using these basic principle on the animals. So first is quantification of exposure, justification of the time point, which is very important, I will discuss. Setting of dose, how you can select the dose, what is the dose setting method, low dose, intermediate, high dose, extent of exposure, how you can choose the drug will be exposed for acute, chronic or subchronic toxicity on the animals, complications, route of administration and determination of some metabolite and statistical analysis for the, you can say, uh, what are the result which you are getting after the statistical analysis. So first thing which is very important, that quantification and extent of exposure. So you know that whenever you will administer any drug to rat, to mice, to guinea pig, to rabbit, to dog, to any other animal, you know that the basic principle that if you want to check the concentration of the drug in the body of animal, it always be from the AUC, that means area under the curve. That means you calculate C max and T max, so maximum concentration. So the basic concept, many a time the student ye ask that how we can go for this. At least we should go for the 3 time replication of the T half of the drug. Suppose if your drug T half is 3 hours, so you go multiply by 3 and you go for the analysis at least for 9 hours in the plasma of the animal, so that you can go for the better prediction that whether what was the maximum concentration of the drug in the blood of that animal. The second thing which is very important, that there could be some species difference, like you can say dose group or some sex. Yes, it is very important. I will tell you one thing, that if you will use a rabbit, the rabbit easily develop a hypercholesteremia, hyperlipidemia instead of rat. The reason is that rabbit is unable to excrete these fatty acids from the degradation by the liver and excretion from the fecal matter or by any other route. So that's why in case of rat, if you want to induce obesity, it will take a more time as compared to rabbit. So this is a little bit difference between a species, and it can be a sex dependent. The second thing, the toxicity study the exposure and the dose dependence in the human at the therapeutic dose level. So this is a big question, that sometime there is a one question in the mind of the researcher, that how you can select a particular dose and what will be the exposure time of the dose. So simply you can see that the dose which you are selecting should be from the therapeutic dose. So suppose if you are giving a 500 mg of paracetamol to the human being, so you can calculate as such. In case of animal, suppose its body weight is 200 gram, so you can calculate that if this is for 70 kg human being, you are giving a 500 mg, then how you can give it for 200 gram of animal? Then you, once you calculate, you multiply it by factor 7, and the dose which you want to administer you can easily find out. So that means basically the factor is there. For the rat it will be 7, for the mice it will be 10 or 10 point something. So these factors are there, how you can calculate the human dose toward the animal dose. But likely you can see that if you want to perform toxicity studies on the animal, then you should know that the exposure of that drug which you want to analyze should be equal that has been reported in the humans. So suppose if you are giving amoxicillin to the human being maximum for 7 days and not more than 10 days, so you cannot expose another derivative of amoxicillin toward animal for 1 month, 3 months, like that. So end point should be similar like a human being. So this you should kept in mind. So this is the species difference that can change the pharmacodynamic parameter of the drug. So what will be the second point? This is very important, sampling point. So many a times the other departments like pharmaceutics, biochemistry or MSc students, if they are doing their projects on the animal, so many times they didn't know that how much blood of a animal we can take. So it is very important that you cannot take more than 10% of the total circulating volume. This is also applicable in humans. So suppose if you are donating your blood to anyone, if you will, you can say donate more than 10%, it can cause hypovolemic shock, it can cause hypoxia, it can leads to cardiovascular problem like that. Similarly, in case of animal, suppose if you know that there is a animal with a body weight of 200 gram, so we assume that the rat will be having a total blood volume of 15 ml. So you keep it 10%, not more than that. So the 10% volume of this will be not more than 1 or 1.5 ml for a 24 hours. So that means you should keep in mind the sample size which you are doing for this should be around per day not more than 0.5 ml, and always not be more than 1 ml. So you should keep in mind this thing, that suppose if you are doing pharmacokinetic analysis, because you have prepared one formulation and you want to do pharmacokinetic analysis, suppose you are doing your work in pharmaceutics or any pharma analytical, either you are doing in industry, so what you will do there, you will do your sampling at zero point 0.4 minute, at zero point 0.4 hour, 1 hour, 2 hour, 4 hour, 8 hour, like that, up to 24 hour. So you should firstly calculate that the blood you are taking should not be from the same animal. because if you will take 0.5, 0.5, 0.5, it will